I'm Andrea Ventura. I'm an MD, PhD. I'm an associate member at Memorial Sloan Kettering Cancer Center, and my work studies cancer using cutting-edge new technologies. The real revolution that has happened over the past 20 years is we have developed tools to sequence the genome very, very quickly and very cheaply. And so now we can basically sequence every new cancer patient and know exactly what the genetic makeup of this tumor is. But one challenge with this approach is that now you have these tumors that have hundreds of mutations. And when we have to sort of figure out which of these mutations are important for that particular tumor, because then we can use drugs against that particular mutation. But this is not easy. It's like finding a needle in a haystack sometimes. So there's a huge class of mutations that we call chromosomal rearrangements that are very important in cancer, and we couldn't really model. And so what my lab has done is figure ways to model these complex genetic events very simply and very quickly. And we can do this in cells, and we can do this in vivo directly in mice, for example. And so if we find a patient that has a specific chromosomal rearrangement, what we can do is take these tools we have developed and model the same event in a mouse or in cells and see what happens. And then we can test the drugs before we give the drug to the patient. And so this is really accelerating discovery. We are using a system that's called CRISPR-Cas9 system. And what it does, we can go into specific position in the genome and clip it. We can find the one we want to attack and do a specific cut there. This is incredibly effective, so you can do very easily. And so the basic idea is that if we do two cuts at the same time in two different places, what happens to the cell with some frequency will try to repair these two cuts. And sometimes we'll swap what's in between, or we'll fuse the position where the two cuts happened, and so reshuffle the genome. By deciding where we do the cuts, we can model what happens in cancer patients. Now we can do this in weeks. We can go from a patient, find what the mutation is, and within a week we can model this in mice or in cells. It's incredibly faster. If you are unfortunate you have cancer, most likely what they will do is take the tumor and sequence the tumor. Knowing which mutations are present in the tumor might tell the doctor, okay, this tumor will likely respond to this specific drug. This is what we call precision medicine. But many patients, unfortunately, don't have mutations in the common genes. And so the goal of my lab is to quickly model them and test drugs that eventually we can give to the patient. The ability to make these mutations fast and to test this hypothesis in a matter of months rather than years, it's really the innovative aspect of our research. And the beauty, I think, is that now the tools we have allow us to address these questions. And it's a very exciting time.